In this screencast, I'm going to speak a little about riboswitches. I'm going to give a general overview about what they are and how they regulate gene expression in bacteria. By definition, riboswitches are sequences at the 5' end of some mRNAs, which regulate the expression of the gene associated with that mRNA through binding to a specific effector molecule. And I'll explain what an effector is a little bit later. They act kind of like a switch. When the effector is bound to the riboswitch, it changes the conformation of the 5' region, which either turns on or off the expression of that gene. Changing the conformation of a riboswitch results in transcription or translation attenuation. And attenuation is the premature termination of either of the two processes. Of all attenuation mechanisms, riboswitches require the least amount of energy and work the fastest. They are a quick way for bacteria to respond to stress conditions. And overall, about 4% of bacterial genes are controlled by riboswitches. Riboswitches show binding specificity to certain molecules called effectors, which can be both extra or intracellular in origin. The concentration of effector molecules within the cell is therefore related to the attenuation of specific genes. On this slide is an up-to-date list of all common riboswitch effectors. Genes regulated by riboswitches are usually involved with metabolite biosynthesis and transport. A metabolite is a molecule involved with a bacteria's metabolism. Most riboswitches are involved with something called negative feedback loops. For example, the lyc gene, which encodes for lysine. After lysine reaches a threshold level within the cell, it can shut off its own production by binding to riboswitches. There are two sections which make up a riboswitch. The first is the aptamer domain, which is the section that binds to the metabolite. It is usually between 30 and 150 nucleotides long, and is the most conserved structure between different species of bacteria. The expression platform contains the mRNA that encodes the gene. In response to effector binding, the expression platform will change shape, inhibiting or helping the expression of that gene. Now I'm going to describe some mechanisms in which riboswitches turn on or off gene expression. The most common is through premature transcription termination. If a ligand binds to an aptamer while the mRNA is in the process of being transcribed, then the riboswitch will change conformation and form a transcriptional terminator, causing RNA polymerase to fall off the mRNA and stop transcription. A less common mechanism is transcription initiation. In this process, instead of creating a terminator, the effector binding will stabilize an alternative anti-terminator and allow transcription to be completed. Anti-terminators and terminators are mutually exclusive RNA conformations. Most 5' UTRs naturally assume an anti-terminator structure before binding. Here's an animation to better describe transcription termination. Initially, the RNA is in a stable anti-terminator loop, and the expression of the gene is on, so RNA polymerase can transcribe the entire strand. When a ligand is bound, the expression platform forms a terminator loop and prevents RNA polymerase from completing its job. In addition to transcription, riboswitches also control translation attenuation. To terminate translation, the aptamer will bind to a ligand and change to a conformation which inhibits the Shine-Delgarno sequence, or the ribosomal binding site, so the gene is never able to be transcribed. Translation initiation occurs when effector binding changes the formation of an anti-sequester hairpin, allowing translation to proceed. An anti-sequester hairpin is only created if the ribosomal binding site is already naturally inhibited before binding to an effector. Some select riboswitches regulate genes by alternative mechanisms. For example, the GLMS riboswitch cleaves and degrades the mRNA, instead of using transcriptional or translational attenuation. Instead of regulating only genes involved with metabolism, some riboswitches turn on or off genes associated with motility and virulence. An example of a riboswitch controlling motility is the cyclic di-GMP riboswitch. Thermosensors are regions of 5' ends of some RNAs similar to riboswitches, but instead of responding to effector concentration, respond to changes in temperature. Thermosensors are often involved with turning on or off virulence genes at proper extracellular conditions. Sometimes a riboswitch will be responsive to more than one effector molecule. Such as the glycine riboswitch, 2 molecules are needed for the riboswitch to regulate the gene. Sometimes other environmental signals, such as pH level or temperature, can affect a riboswitch and work in tandem with effector binding. On this slide is an up-to-date chart of the known riboswitches. Riboswitches are first categorized by the ligand they bind to, which is called the family, and are then separated into classes by the secondary structure of their aptamer domain. In addition, this chart also says a little bit about the types of genes in each family that each family regulates, as well as the dissociation constant for the bound effector aptamer structure. This KD value, as well as the regulation mechanism of the riboswitch, determines how the riboswitch is controlled, which is something I'll explain next. A recent controversy in the riboswitch world has been on whether or not riboswitches are kinetically or thermodynamically controlled. Kinetically controlled means the concentration of the ligand is the driving force in the reaction, and the final products do not reach full thermodynamic equilibrium. Thermodynamic control means the energy stability of the products is the only driving force. If a riboswitch controls genes through transcriptional attenuation, it is believed to be under kinetic control, because the binding must be fast enough to compete with the RNA polymerase. For this to be so, a higher concentration of metabolites must be present in the cell relative to the KD value of the binding. If a riboswitch regulates through translational attenuation, the speed of the effector binding is not as important as the stability of the product. So generally, these riboswitches are under thermodynamic control. To conclude, riboswitches are very important for gene regulation. They allow for rapid response to environmental stresses, which is necessary for the bacteria's survival. Because of this, riboswitches are currently being researched as new targets for antibiotics. Not only would antibiotics targeting riboswitches not affect human cells, but aptamer domains are highly conserved, so there are possibilities for the antibiotics to be effective in broad spectrum. Finally, riboswitch research can aid in our knowledge in how bacteria adapt and become resistant to certain environmental conditions. Mutations in the aptamer domains of riboswitches can lead to alterations in important gene function regulation. And here are my resources.